Hello, everyone. My name is John Semple, and I'm a professor of transfusion medicine at Lund University. And I'm going to talk to you today about the pathophysiology of immune thrombocytopenia. Now, immune thrombocytopenia, as you probably know, is an acquired autoimmune bleeding disorder. It has an incidence of between 3 and 5 per 100,000 and a prevalence of 9 to 11 per 100,000. And the nomenclature for ITP was changed about 10 years ago. It is now simply called immune thrombocytopenia. The purpura has been dropped because not all patients present with purpura. It's categorized as an isolated thrombocytopenia of a platelet count of less than 100 times 10 to the 9th per liter. And there's 4 new diagnostic criteria or categories that are associated with ITP. Newly diagnosed ITP is within 3 months from diagnosis. Persistent ITP is between 3 and 12 months from diagnosis. And chronic ITP now is reserved for those of patients that have ITP for greater than 12 months duration. Severe ITP is the presence of bleeding symptoms, either at presentation or at any time during the course of their disease. Now, the classic immunopathogenesis of ITP is antibody-mediated. Autoantibodies are generated against platelets. They bind and opsonize the platelets, and as the platelets traverse through the spleen, they're very quickly destroyed by macrophages through Fc receptor-mediated phagocytosis. And this is still considered the primary mechanism of how platelets are destroyed in ITP. Now, with respect to the autoantibodies that are generated in ITP, they can be categorized based on their specificity and their classes. Now, this is if you think about it, this is the footprint of all the autoantibodies. Interestingly enough, about 10 to 20% of patients, even though they're diagnosed with ITP, they are in fact autoantibody negative. About 20 to 30% of patients have mixed specificity antibodies, which means that they not only bind to one particular glycoprotein, but they bind to other epitopes, either on that glycoprotein or others. And this mixed specificity are of IgA and IgM isotypes. But the predominant autoantibody footprint in these patients, about 50 to 60% of them also have mixed specificity antibodies, and they're all IgG1 and 3 dominant. And this is really the IgG antibodies that are really the main autoantibodies that generate the increased phagocytosis of the platelets. Very few of the patients have single autoantibody specificities. That's very important. Most of them present with multiple specificities. And of course, we can't forget that underlying all of these autoantibodies, in some of these patients, or perhaps most of them, there are also CD8 positive T cells that can also mediate platelet destruction. And of interest, these CD8 positive T cells may be insensitive to certain therapies. And we'll talk about that in just a moment. Now, it's interesting, these 20% of patients that are negative for autoantibodies. Why is that? Is it because the tests are not robust enough? Or is it that they're truly negative and that there is a T cell involved? Well, in 2003, Hans Wadenwijk showed very nicely that in patients that are autoantibody negative, during active disease, they could take their peripheral blood mononuclear cells and they could show that they had T cells that could lyse platelets in vitro. And when the patients went into remission, either spontaneously or during therapy, in fact, this cytotoxicity went away. So this was the first demonstration that in addition to the autoantibodies that destroy platelets, T cells, CD8 positive T cells, can also mediate these effects. Now, we now know that the low platelet counts is not just because of peripheral platelet destruction, but in fact, the immune response against platelets can certainly migrate into the bone marrow and inhibit or destroy megakaryocytes within the bone marrow. And on the top is electron micrograph of a megakaryocyte on the right from a patient with ITP, and you can see that the cytoplasm has become vacuolated. and the nucleus has condensed compared to a healthy megakaryocyte on the left. This is antibody-mediated ITP, but with CTL-mediated ITP, you get very similar effects. You can see on the right, these are mice that have CTL-mediated ITP. You can see the macrophages are quite abnormal, and they're undergoing what appears to be apoptosis. And in fact, in both of these studies, there were apoptotic markers that were found associated with the megakaryocytes. Now, there's a number of ways that platelets can be destroyed, and we know many of them now. Just in the middle here, in the spleen, for example, what I just told you, FC receptor-mediated phagocytosis can take place. And there's also T cells that can also destroy platelets directly within the spleen. And we know that it's probably got to do with a lack of tolerance. There's a T regulatory defect, and we'll talk about that in a moment. In the blood, there is certainly, the platelets are opsonized by IgG antibodies, and these are complement binding autoantibodies. And certainly it's been shown that complement can in fact bind to these opsonized platelets and lyse the platelets right directly within the blood. And also there's an interesting aspect. Some antibodies can change the platelet phenotype. They can desilylate the platelets and strip off sialic acids. And when this occurs, it's thought that the platelets are then destroyed, not within the spleen, but by hepatocytes within the liver through the Ashwell-Morel receptor. And finally, as I mentioned before, in the bone marrow, both antibodies and CTLs can migrate into the bone marrow and cause destruction of the megakaryocytes within the bone marrow. So we now know that there are multiple sites and there are multiple mechanisms of platelet destruction within ITP. Now I want to move on to some very recent data that I think is quite important, and it's really the effects of platelets themselves in ITP. Platelets are not just innocent bystanders in this disorder. We know from a large literature that platelets are actually immune cells also. Besides their hemostatic function, which is probably their main physiological function, they have a lot of different immune molecules associated with them. And I'm not going to go through all of this today, but I'm just going to go through a few aspects of this and how the platelets themselves may be able to regulate the autoimmune attack against them. Just a review of T helper cell subsets. T helper cells, the CD4 positive T cells, can differentiate into various subsets. Some of these you've probably heard of. A naive CD4 positive T cell is acted upon by antigen presenting cells, and depending on how the antigen presenting cells deal with the naive cell, they can differentiate along four main pathways. Th1 and Th17 cells are very pro-inflammatory cells, and they're very associated with autoimmunity and inflammatory disorders. Th2 cells are anti-inflammatory, and they tend to counteract the effects of Th1. And of course, we now know that T regulatory cells are very immunosuppressive cells, and they're responsible for immune tolerance. And we now know that, in fact, they are defective in patients with chronic autoimmune diseases, and that includes ITP. Excuse me. If one looks at the Treg literature within ITP, it's quite a robust literature. And the interesting thing about all of these papers is that they show almost the same thing, that Tregs are either lowered, it shouldn't be elevated, it should be lowered in number or function during active disease. And this causes a lack of tolerance, and this allows for pro-inflammatory responses. The literature also talks about the Th17 responses. And in fact, these show that in fact, during active disease, these cells and Th1 cells are elevated in number and function during active disease. And this allows for a relationship to be generated. And you can see that on the left of this graph, when platelet numbers are low, there's a defect in the Tregs and cytokines associated with Tregs, such as TGF-beta and IL-10. And this allows for pro-inflammatory Th1 and Th17 cells to become activated and drive autoimmune responses against the platelets. Now, as the platelet counts go up, and it doesn't matter how they go up, it appears, all these different therapies that increase the platelet counts in patients with ITP have shown the same effects. What happens is you get a rescuing of the Treg numbers and Treg functions within ITP, and this correlates with an increased immunosuppressive cytokine response from these Tregs. And this suppresses the Th1 and Th17 responses that are driving the autoimmunity. So this is actually a very interesting relationship because it suggests that all these different therapies, for example, have very different mechanisms of action. It may be that just simply increasing the platelet mass is enough to change the autoreactive nature of the patient. And we're going to talk a little bit more about this, and this may very well be to the fact that platelets are immune cells. One example of how platelets themselves can affect the immune response is this very nice study that was published by the Nurden groups in France, showing that in fact platelets, because they contain the autoantigen, in this case 2B3A, GP2B3A, they also express CD40 ligand, CD154. And this is a co-stimulatory molecule for autoreactive B cells in general, and in this case for autoreactive B cells. And this group showed that if you mix activated platelets with autoreactive B cells from patients with ITP, these platelets can actually directly drive these autoreactive B cells to differentiate into plasma cells and to generate 2B3A autoantibodies. This suggests that perhaps the platelets themselves have a role to play in how antibodies are generated in patients with ITP. Now we're going to talk and switch gears a little bit and talk about antigen-presenting cells, because chronic ITP, like all organ-specific autoimmune diseases, is really a T-cell mediated disorder. You need these T cells to generate autoantibodies, and antigen-presenting cells are critical in turning T cells on. Well, there's been quite a bit of work done with dendritic cells in ITP. Dendritic cells are the most potent antigen-presenting cells in the immune system. And what these studies have shown that in fact, dendritic cells within patients with ITP have a reduction in an enzyme called indolealamine 2,3-deoxygenase. And this is the enzyme that catalyzes tryptophan to kenurein synthesis. And because this enzyme is become lowered in the dendritic cells, it increases IL-12 production. And this actually reduces the Treg response, but it increases pathogenic T cell activation. So this is a very good example of how abnormal responses from dendritic cells can drive T cells into an autoimmune phenotype. Now we're going to switch back and still talk about antigen-presenting cells, but now let's think about platelets acting as antigen-presenting cells. The one interesting thing about platelets and one of their immune characteristics is that they have all the capabilities of processing and presenting antigens and loading these antigens onto MHC class I molecules. They contain an intact proteasome, which will degrade denatured and autoantigens potentially into peptides. They have all of the necessary proteins within the alpha granules that can translocate these peptides into the alpha granule and load these peptides onto MHC class I molecules. When the platelet becomes activated, the alpha granules are exocytosed and the class I molecules are also expressed. And these class I molecules can then theoretically interact with CTLs in lymph node, in the bone marrow, and perhaps in the spleen. This was very nicely shown by Craig Morell's group, who showed that in fact platelets could present a variety of different peptides to CD8 positive T cells. Interestingly enough, they showed that platelets could present plasmodium peptides to CTLs because platelets, as I told you before, have co-stimulatory molecules, so they can actually act as an antigen-presenting cell and cross-present these antigens to CD8 positive T cells to cause them to activate, and these T cells can then go in to target and kill target cells that are infected with malaria. This has now been shown for a variety of other proteins, including self proteins such as 2B3A molecules. Platelets are very good at producing extracellular vesicles. They are probably the biggest source of extracellular vesicles within the peripheral blood. And Genevieve Marceau in Eric Bulliard's lab beautifully showed that, in fact, when platelets activate, they can release these platelet extracellular vesicles. And the interesting thing about these extracellular vesicles is they also contain the proteasome. It appears that the platelets actually transport the proteasome into these extracellular vesicles as cargo. And this allows these extracellular vesicles to then process and present self-antigens to CD8 positive T cells. Now, platelets themselves are locked within the circulation. The only lymphoid organ that they can see is the spleen. The only way they can move out of the circulation is if there's damage of the endothelium, and then they can move into the tissues. But what they showed is that these extracellular vesicles can readily move into the lymph nodes of animals. And in fact, it's really important, I think, this finding, because it may be that these extracellular vesicles from platelets might be responsible for generating CTL responses within the lymph nodes of both mice and patients with ITP. We've also shown from our group that, in fact, the platelets are really derived from the megakaryocytes within the bone marrow. And in fact, megakaryocytes have an active proteasome, and they can process and present not only foreign peptide antigens from exogenous foreign protein antigens, but they can also process and present self-antigens. They can load these peptides onto MHC class I molecules, and they can actively present these peptides to CD8 positive T cells. The interesting thing about this process is that at the same time that they're presenting and activating T cells, they're making platelets, these megakaryocytes, and they can load these MHC class I complexes into platelets. And this might be a way for them to communicate with the peripheral immune system in the spleen, for example. So ITP pathophysiology has become quite complex. It's thought that, in fact, the reason for the whole aspect of this is that there is a reduction in T regulatory cells, a lack of tolerance. This allows for macrophages within the spleen to present platelet autoantigens to T helper cells. These drive B cells to differentiate into plasma cells to generate autoantibodies. These also can drive and activate CD8 positive T cells that can migrate into the thymus and destroy megakaryocytes. And of course, when these autoantibodies are generated, complement proteins can be activated on the surface of the platelet, and they can destroy the platelets intravascularly within the blood. And if we look at all the various therapies for ITP, these therapies appear to target some of these processes. And these are new therapies up here, FcRn, the neonatal Fc receptor, Syk inhibition, and that they seem to target the macrophages primarily. Rituximab targets the B cells and removes the B cells. There are now new therapies on complement inhibition that are being looked at in ITP. And of course, the thrombopoietin receptor agonists drive megakaryocytes to generate more platelets, and they increase their resistance to apoptosis. And there's a whole variety of therapies that are coming down the pipe that I think will be available to patients, for patients with ITP in the next few years. So in conclusion, the pathogenesis of ITP has become a very complex phenomenon, and it involves almost all elements of the innate and adaptive immune system. Abnormal Th1 and Th17 responses are activated during active disease, and this is due to a lack of Tregs, and this is a lack of tolerance. And it's really interesting that platelets, because of their ability to be immune cells and modulate immune responses, they may in fact be very important and responsible for modulating autoimmunity and ITP. They may be dictators of their own fate, if you will. And of course, further research is required to really determine these precise immunopathogenic mechanisms that modulate platelet counts in ITP. And I'd like to thank you all for your attention. I'll be glad to answer any questions. Thank you very much.